Can everyone see my screen?
Yeah.
Great, okay. So a couple of announcements. Quiz number 7 will be due by this weekend on Saturday night. I will have quiz 8 out. I'm hoping to make it not too hard. It won't be, I promise. If you haven't uploaded your presentation, please upload it to Blackboard. I don't put in your grades until you have submitted that PowerPoint on there because it won't let me give you a grade otherwise. So please do that. Also, it's for my records too that you guys actually did present. I can be like, yes, this person presented. PowerPoint. Here you go, you know? So make sure you do upload that if you haven't already. Whoever's presented so far, I guess. We don't have anything else. We can go into presentations. Who wants to go first?
I can go first.
Okay. Oh, wait, let me enable sharing. Okay.
Can you see it?
Yep.
All right. So my question was, the condition that exists when an organism gains or loses one or more chromosomes, but not a complete haploid set, is known as, and it's one of these 5. And then the answer, oh, let's see. The answer is aneuploidy. Sorry, it's blocking it off. All right. The answer is aneuploidy because definition simplified is an abnormal event where you either gain or lose a single chromosome. The other event, triploidy, means a third duplication where you would see 69 chromosomes in a human. And then polyploidy means to just have more than one chromosome. And similarly, a euploidy just means normal duplication. And then trisomy closely resembles the correct answer, but is incorrect because it's a type of aneuploidy where you only gain. Yeah, that's about it.
Does that make sense to everyone?
Yep.
Awesome. Okay. Are we missing people who has to present?
Mine was from last week, by the way. It was supposed to
I
be
know.
last week. Yeah.
So remember, guys, from last week is pushed this week. So I have Shania and Quentin on my list for last week or this week. Whichever day you want to go with.
I was supposed to present 2 weeks ago, but I could present now.
Okay.
Okay. Should I share my screen?
Sure.
Can you guys see it?
No.
Okay. So my question was about chromosome mapping, and it was 2 plants, both dominant AB and recessive AB, whose loci are linked together and 35 map units apart. What will the proportions of the following gametes from a single plant be? And it's supposed to be the dominant AB, recessive AB, recessive A, dominant B, dominant A, and recessive B. And the answers that I got are, for dominant AB, it's 0.175. For recessive AB, it's 0.175. And for recessive A, dominant B, it's 0.325. For dominant A, recessive B, it's also 0.325. And basically, the genotype produces 5 4 types of gametes. For the recombinant gametes, it's 35% in total, which makes up the 17 for each. And for the non-recombinant is 65%, which is the 32.5 for each.
Yep. Everyone cool with that question?
Yep.
Okay. Shania, are you still presenting today?
Yes.
Okay. I don't see Quentin on here yet. So would you like to go?
Yes, 1 second. I'm just trying to share the screen.
Okay. Everything okay?
It's not allowing me to share my screen.
That's weird. What is it saying? Is it saying something specific or?
It's saying that I have to go in my settings in order for my computer to grant access.
That's weird. Okay. Let me try something. If I can find your name, that will help. Okay. Oh, perfect.
Can you guys see my screen?
Yep.
Oh, okay. How do I make this bigger? Okay, so my question was a human female with Turner syndrome also expresses the X-linked trait homophilia, as did her father. Which of her parents underwent non-disjunction during meiosis, giving rise to the gamete responsible for the syndrome? So we know that the X chromosome comes from the mother, and the male is XY and the female is XX. Males will be carrier, females will have the syndrome, and only one of the X became infected. And the answer is that non-disjunction occurred in the mother. This is because the only X chromosome from the daughter must have come from the father. It could be either primary or secondary non-disjunction.
Good. Okay. So I haven't heard from Quentin, so we'll just continue for now. Okay. So today's session, I kind of have it in 2 parts. We're gonna go over some questions from chromosomal mutations, and then we're gonna go into a little bit of an overview, I guess, for the genetic code and central dogma. just enough to get you guys through the presentations next week, because I realize the sessions themselves are kind of a the presentations are a little bit of a head lecture. So I just want to make sure you guys have enough info that you guys can answer the questions properly. Okay. Let me see if this thing lets me. Okay. So chromosomal mutations, they come in two different types. One is completely structural, and then the other set is more variations in the number of chromosomes, okay? So we're gonna go through each of the different structural abnormalities first. So let me just get, can everyone see my pointer?
Yeah.
Okay. So what do you guys think this is? So it's saying that, so we have a region of DNA chromosome just X'd out, and the chromosome became
Is
shorter.
it deletion?
Deletion.
Yep. Yep. Good work. Okay, so that's deletion. What about this guy? We have one region that's put twice.
Duplicate.
So this
Duplication. Sorry.
Yep. What about this guy? So the ends are following the arrows here.
Cross.
If you guys can, huh?
Is it crossover?
So crossover is tiny regions. This is like an entire section of a chromosome. So wait, somebody said something. What happened?
Is it inversion?
Yes. So essentially inversion is a region that is taken and flipped over, right? So this is not two different chromosomes. This is the same chromosome where a region of DNA is flipped, region of the chromosome flipped 180 degrees. Does that make sense?
Yeah.
Okay.
Yeah.
Oh, okay. This one wasn't that obvious, but this is kind of substitution where a region of a chromosome just kind of goes in and be like, okay, so this is also called non-reciprocal translocation, but it's also called substitution. Okay.
Okay.
Then this guy, what do you guys think this is?
Reciprocal translocation.
Yep. So there's non-reciprocal versus reciprocal translocation. Non-reciprocal is only one way. Reciprocal translocation is both ways. Okay. Great. So with inversion, there are two types of inversions. One is called paracentric and one is called pericentric. So in this case, it's asking the differences between those and an example of each. My previous class said that you guys did not cover this in lecture yet, so I'm gonna go through it. Unless you guys want to take a stab at it. What do you guys think when you hear paracentric and pericentric? Yes. Okay. So that is correct. Okay. Okay. So paracentric, it does not change the length of the two arms of the chromosome, and the centromere is not involved. That's why the change, the lengths don't change in paracentric, right? So we have a chromosome. Can everyone see my chromosome? So here we have the chromosome, we have the centromere, and the change is occurring. The inversion is occurring away from the centromere, right? So it's not involved at all. Whereas with pericentric, it changes the length of the two arms of the chromosomes, and the centromere is actually involved in the inversion. So we see it here. It's like, okay, we have the two changes that we have. So the changes we have of the inversion, and we see that the centromere is completely involved. Does these two inversions make sense?
Yeah.
Sometimes my internet is being crazy here, so if you guys can't hear me, let me know. Okay. So next we have chromosomal mutations that involve the variations in numbers. There are two main categories in the number variation set. So the first one is called aneuploidy. Aneuploidy essentially involves like loses a chromosome or two or more, but not an entire set. You can lose or gain in that case, okay? Then there's something called euploidy. With euploidy, it's multiples of the haploid set. So if you have an extra haploid set or anything more than that, that's when you get euploidy. So just to kind of go through this, aneuploidy is the diploid number plus or minus any number of chromosomes, right? So X chromosomes. Don't confuse them with sex chromosomes. I hate that this figure has it as X chromosomes. It's the variable, okay? Not the sex chromosome. So an example would be like monosomy. Monosomy is one less than the diploid number. Disomy is just another word for diploid, which is also 2n, okay? Then we have trisomy. Trisomy involves an additional chromosome. So that's why it's labeled as the diploid number, or 2n plus 1, okay? Then it just goes on from there, like tetrasomy, pentasomy, going like 2n plus 2, 2n plus 3, so on and so forth, okay? Then euploidy, multiples of n, that's like diploidy here is 2n. So one thing with aneuploidy, it's never a complete, it's always one or more, but not an entire haploid set, okay? Diploidy is 2n, like we know. Polyploidy just refers to anything that's like 3n, 4n, anything above the diploid number, okay? Like it's a extra haploid set from 3n onwards, okay? So anything after 2n, 3n, 4n, 5n, 6n, list goes on. That's called polyploidy. Then 3n has its own name, triploidy. You see that in like plants or certain animals. I can't think of examples off the top of my head right now. Then it just goes on tetraploidy, pentaploidy, etc. So 4n, 5n goes on. Any questions so far?
Nope.
Okay. Autopolyploidy. So that is essentially multiples. So these are involving the ploidy issues. It's so autopolyploidy ends up with multiples of the same genome. So 3n and above for the same genome. Whereas allopolyploidy or ampidiploidy involves multiples of closely related genomes. Okay? So that's just differences in terminology. Okay. So here we have a question.
Can you go back for a sec so I could just take a quick picture? Thank you.
I'm going to be posting it online too. Don't worry.
Thank you.
Okay. The haploid number in a certain species is 10. How many chromosomes are present in a cell from a triploid member of this species? Yes, okay. Anyone else has an idea? I got the correct answer so far. Do I need to bring back the table?
Is it 3? Is it 30?
Yep. So, oh, come on. Okay. So, as we know here, triploidy is 3 times the haploid number. So that's how it becomes 30, because you have a haploid number here of 10. 3 times 10 is 30. We cool?
We cool.
I like to hear that. Okay. Which of the following genetic abnormalities is most likely to result in viable offspring in humans? Okay, so oh, did somebody say something?
Is it 2?
Yep. So trisomy 21, what does it cause? If this thing lets me, okay. So what does it cause? What disorder? Do you guys know?
Down syndrome.
Down syndrome, yeah.
Yeah, you guys are good. Okay. Which of the following embryos is least likely to survive live birth? Do you guys know?
I want to guess 4 because I feel like the bigger the chromosome, the more important it is. But I don't know.
Okay, you're close that it is choice 4. The reasoning behind this is that with autosomal chromosomes, you have a lot of critical genes that you need for survival, right? So if one of the chromosomes are missing, some genes, they need the multiple alleles or genes that are of the same trait to work together to actually show that trait, right? So that's why if you have monosomy for chromosome 3 is an autosomal chromosome, and it could be, and it does have different chromosomes, not chromosomes, genes that you need that are critical. So that is what, so monosomy in humans is lethal. Okay. Do you guys know what this, so we know monosomy X is Turner syndrome. What do you guys, so what is this guy? What does this translocation cause? Similar to this guy. Actually, it's the same thing. Well, oh, wait, say that again.
Down syndrome.
Yep. So those are the two ways you can have Down syndrome. It's the translocation between 14 and 21 or trisomy 21, okay? How is the chromosome complement in an individual with monosomy written?
Is it 5?
Yep. So monosomy, just one less chromosome. And as we can see in our table here. Good. Which of the following terms refers to a failure to separate a pair of homologous chromosomes during anaphase 1 of meiosis?
No.
Wait, Elizabeth, did you say non-disjunction?
Yeah.
Okay. Yep. So that is the answer. This is the definition for it. Okay. Which inherited disorder can be produced by a Robertsonian translocation? So a Robertsonian translocation is like translocating between chromosomes.
Is it familial Down syndrome?
Yep. So that is essentially that's an example of Robertsonian translocation. Does that make sense?
Yeah.
Okay, this question gives away the answer and I completely forgot. But anyways, a small deletion in the small arm of chromosome 5 is known to cause blank syndrome. So you see that part of it's cut off in the second chromosome. Crazy shot, right? Forgot that I had the answer here. Do you know why it's called crazy shot?
They sound like a cat crying when they're born.
So essentially what crazy shot means is a cat's cry. It's because infants with these syndromes actually cry in the sound of a cat. So they called it cat's cry syndrome in a sense. It just sounds fancier calling it crazy shot. Oh, wild scientists, I tell you. Okay. Which of the following genetic variations is not an example of aneuploidy? What do you guys think?
Is it 2?
Yep. Yep. That is 2. So, as we all remember, aneuploidy is either gaining or losing a couple of chromosomes, but not an entire haploid set, right? Cool. So this next part is kind of more of an overview, just like talking about the dogma, differences between prokaryotes and eukaryotes in this situation, and we're going to talk a little bit about the genetic code table. Okay. So if you guys don't remember anything from genetics, please remember this. Okay. I will get yelled at by the molecular bio TA because they have questions on this, and this is one of the basics that we have to remember. So the central dogma of biology here, we'll see that, okay, so there is a lot more to this process than what is just shown here. There's a lot of factors, a lot of things. But for this class's purposes, we're gonna stick to the basics, okay? So in this case, DNA can replicate itself to make more DNA. And then through a process called transcription, it can make RNA. And then RNA can either go back to being DNA through reverse transcription, or be go through translation and make protein, okay? So the process of all of this occurs differently between prokaryotes and eukaryotes. Mostly because with eukaryotes, since they most are including a nucleus, they also add in introns. So they kind of add another step to this. Whereas with prokaryotes, you do not see introns. So in this case, and since you do not have a nucleus there, you can actually do them all together, okay? So it can go from DNA to mRNA and protein in one single go. While you can go from mRNA to protein while DNA is still being transcribed into mRNA, okay? Whereas with eukaryotes, you cannot do both of these together because you have a nucleus going on. And since there are introns, it adds an additional step before it gets exported out into the cytoplasm. And then you see that with the mRNA. So there are a couple of steps in eukaryotic cells with RNA processing, because so when it transcribes from DNA to RNA, it still encodes those introns. We do not know the exact function of introns, but we know that they do not code for the proteins itself. So to make it more efficient in the process, the cells themselves splice them out. So they cut it out and they cap it and protect it, and then send it off on its way out of the nucleus into the cytoplasm to make proteins with the ribosomes. We'll go more detail next week in whatever is happening, but does this make sense with the differences? Or
Yeah.
did I lose everyone?
Yeah, that's good.
Okay, I'm happy. Okay. So the genetic code table. So this is kind of how they get from the mRNA stage to protein. So it's essentially a key to translate from RNA, the language of RNA, to the language of protein, okay? So these three nucleotides here, they're called codons. When these codons are together, they come in, so I'll go more detail into this, but this is just essentially these codons code for a specific amino acid. And then, so you can see that this So some amino acids, they end up being coded by multiple different codons. So to read this table, if you have like AUG or this guy, for example, this is also the first one you would do. This is the start codon, okay? The start codon is always starting with, and it's attached with methionine, okay? It always attaches with that. So to realize that if we don't remember AUG off the top of our head, what goes with methionine, you can easily go, okay, so we have AUG, that's our sequence. So we look at the first letter, which is A, right? Then you have U. So first letters on the left-hand side, second letter on the top, third letter on right-hand side, okay? So when we go AUG, you see A, U, and then G, right? So we come down here. Does that make sense in how to read these tables?
Yeah.
Okay. So we have that. Now, so as I said, this is the start codon and everything. Oh my God, the highway's distracting. Okay, sorry about that. Next, we have stop codons. This is when everything gets terminated. These do not code for any amino acids. It just stops it and then lets the protein go. That makes sense. I promise it will make more sense next week when we go through transcription and translation more, I guess. But does that make sense so far as to what we should know for now?
Yeah.
I'm glad. I think I confused the crap out of my last class. So I'm happy. Okay. Oh, okay. So I really got to put my animations in correctly. Okay. Which amino acids could result from a single base substitution in CCA? So C, C, A. So that's proline, right? Indicate the base changes that produces each new amino acid. So the way we go about this question is, so for proline, you know that the third nucleotide doesn't do any change. So we can ignore the third nucleotide, right? So the next thing you would do is look at the first two nucleotides, right? So these, when you change the first letter, you get three different types. Obviously, you get these three combinations. And then every time you have a new amino acid, you would actually write that down as well, right? For this question specifically. So in this case, UCA, ACA, and GCA gives out serine, threonine, and alanine. Whereas with CGA, CAA, and CUA, we get arginine, glutamine, and leucine, okay? Does that make sense as to why those are the combinations and why we just don't wanna? So you can add these in as well if they're specifically asking like, okay, so you can add these guys in as well in there. But it's asking for the base changes that comes out when it's a new amino acid. So does that make sense?
Makes sense.
Yay. Okay. So now, so this is a video on transcription. I did not actually play it for my last class, so it's up to you guys. We can watch this video and end for the day, or whatever you guys want to do, I guess. Questions, concerns at that point. So okay. We actually have more than 10 minutes, so let's watch a video. Hopefully you guys can hear it this time. Okay. Can you guys hear it? Someone?
No, I
No?
No.
Wait,
Really?
try it one more time. I just give it 1 second. I think we might be able to. No, I lied. We can't hear anything.
Okay. I guess no videos. Boo. Okay. So, do you guys have any questions, concerns?
No, and I don't want to burst anyone's bubble, but I feel like maybe if you try and search it in like the search engine instead of having it like play as an attachment in your PowerPoint, maybe we might be able to hear it that way.
Okay, let me try that. Hold on.
I have a question actually too.
What's up?
Is there any chance that he curves the test?
That depends on how the class does. I'm not entirely sure if he will. It's
I mean, you should
that's
put
totally up to Dr. Osuna.
you should put in a word though, you know.
Did you guys actually hear that?
Yeah.
yeah.
Okay, then I'm going through YouTube. Okay, let's do this. Can you guys actually?
No, I can't hear it.
Well, Zoom hates me because when I share things, it doesn't like to let people hear things.
Oh,
But
are you wearing? You're wearing headphones, right?
no,
Oh, okay.
I'm
'Cause
not even
usually-
wearing headphones this time because I wanted to show a video. I think it's because the last time, did you guys hear it when I did Kahoot last time?
Yeah.
Okay, so I think it's because I was using a second screen for that. And it
Didn't
was,
you do a video in like one of our first discussion lectures? It was like the first one or the second one, and we could hear it in that lecture.
I know some of you guys couldn't hear it that day, so I'm not sure.
We tried.
Okay. This is why I miss in-person discussions. Because I have all these fun videos that I like to show you guys. But Zoom is being a pain in the neck. Anyways.
Do you have it on your PowerPoint that you post? Because I can click on it and watch it on my own.
Yeah, it's going to be on there either way.
Okay.
Do you guys have any questions, concerns? Is genetics making you guys want to run away?
Yeah.
Yes.
Yeah.
I hope discussion is helping you not to run away.
Everything seems so simple when we go over it in discussion, but as soon as I have to apply it to a test, I have no idea what's going on. Yeah, I agree.
Yeah, I know. It's exam.
Could we
Yeah.
possibly try like a very difficult question like next time? One that might be like not that it might be on the exam, but like one that would be like the same difficulty as one that would be on an exam, because I feel like the questions we go over are very simple and understand, and the same thing for his class. And then as soon as we get into the exams, it's like the application is far more advanced.
Mm-hmm. Yeah. It's just harder to find these questions, but I'll try looking into more. Because like I feel like the upcoming question like topics are more conceptual. In that because like the beginning half is where all these different applications come in. Whereas the upcoming topics like transcription, translation, those are more on the conceptual side. So I'll definitely try to find more questions that are more challenging. I need to go find that textbook again, but
Thank you.
but I'll definitely try to figure out more. I think I actually have a study guide that you guys might be able to use that has like all the concepts going on there too. Any other questions, concerns? Nope. Great. Okay. If not, you guys are free to go.
Thank you.
Thank you.
Have a good night.
You too.